Dear students, in today's lecture we will try to discuss more about cell division in eukaryotes. In previous lecture we have discussed that cell division in eukaryotes could be divided in two major phases. First was the interphase and second was the M phase or mitosis phase. In today's lecture we will discuss in detail about the interphase. Interphase is basically the extended period of growth and development between the cell division. Although little activity can be observed with light microscope, the cell is quite busy. Why cell is busy? Because that cell is busy in the process in the DNA synthesis, in RNA and protein production, and also hundreds of biochemical reactions which are necessary to do the process of cell division. These all take place during the interphase. These hundred of biochemical reactions basically help cell to do the process of cell division in quite a well defined manner. So there are also several checkpoints which regulate the cell cycle by allowing or prohibiting the cell division. These checkpoints, basically like the checkpoints in the M phase, ensure that all cellular components are present and good or in good working order before the cell proceed to the next stage. So these checkpoints are necessary to prevent the cell with damaged or missing chromosomes from proliferating. Defects in checkpoints can lead to unregulated cell growth as issue in some sort of cancer. So interphase has basically two different subphases. First is the G1 phase. That is basically in this phase, basically the cell grows and proteins which are necessary for division are synthesized, and that phase basically typically lasts for several hours, and this is also very critical point which is known as the checkpoint, G1S checkpoint. G1S checkpoint, that checkpoint that holds the cell in G1 until the cell has all the enzymes which are necessary for the replication of DNA. After this checkpoint has been processed, the cell is committed to divide. But in some situations, before going cell into next phase, cell enters into the G0 phase. That G0 phase, cell may exit from the active cell cycle in response to some regulatory signals and pass to non-dividing phase. That is, G0 phase is basically the stable state and maintain a constant size until there are some mechanisms or there is some need of any process of division. So after, if there is the signal that cell is now ready for the process of division, then cell, after completing its G1 phase or after going through the critical point which is G1S checkpoint, then cell go to the next subphase which is known as the S phase. After the G1 phase, the cell enters the S phase, which is also known as the DNA synthesis phase, in which each chromosome duplicates. Although the cell is committed to divide after the G1S checkpoint has been passed, DNA synthesis must take place before the cell can pass to the mitosis. So if DNA synthesis is blocked, or that blockage could be because of any sort of drugs or any kind of mutation, the cell will not be able to undergo mitosis. Before S phase, each chromosome is unreplicated. After S phase, each chromosome is composed of two chromatids. After the S phase synthesis, the cell enters the next phase which is known as the G2 phase, and before entering into G2 phase there is also another important checkpoint which is known as which is the process which is known as the synthesis of the chromosome. So during the G2 phase, several additional biochemical events which are necessary for cell division, they take place. There is another important or critical G2M checkpoint is reached near the end of the G2 phase. This checkpoint is passed only if the cell DNA is undamaged. Damaged DNA can inhibit the activation of other enzymes. So that is very important checkpoint. Although the length of interphase varies from cell to cell type, a typical dividing mammalian cell spends about 10 hours in G1 phase, 9 hours in S phase, and 4 hours in G2 phase. This is basically the overall diagram which is indicating about the overall process of interphase which takes place during the cell division. Here you can see that G1 phase of the interphase, there is cell growth. In S phase there is the process of DNA synthesis. Again in G2 phase there is again the process of cell growth and division. When that G2 phase is complete, then cell automatically undergoes the process or enters in the M phase. Thank you.